Welcome everyone. We'll go ahead and get started. So we're going to kick off in terms of the clinical context for things. And as we're going through this, I'm going to put together my wish list, which is mostly for the families actually, so you guys can help me as we're going through with this. And I'm tag teaming with Rada, so she's going to give also the other part of the clinical update. So correct me if I'm wrong, Carol, but we now know of 64 individuals who are out there around the world in terms of PPP2R5D, and there are also a small number with the other PPP genes. It's a handful, though, if I'm correct.
5 of the 1A and 1 of the C.
Okay, and for those, I'm not showing any data today because we haven't been collecting that systematically. But for the condition I'm going to show you about, Carol and I and Rada, just to give people an update, actually Kim and Rada and I are working on what we call a gene reviews, which is for the families, something that's published online and is a very valuable go-to resource for clinicians. It's very concise and gives an update in terms of what we know about the condition, gives clinical recommendations, and so we are trying to get together as much data as we can to make that as robust as we can. The gene reviews is going to be just about PPP2R5D. We thought about doing something else, but we've eventually decided that's where we've got the most data. We can only report in that on published data, and so we're actually going to have a tag team approach, which is we're going to have a gene review submission, and then we're also going to have a clinical update with all the cases that we can, and actually publish that in, I don't know if it's going to be American Journal of Medical Genetics or something like that, with the idea being that once that's peer-reviewed and accepted for publication, gene reviews is something we update on an annual basis, and once it's published in the scientific literature, we can come back and bring that into next year's updates on the gene reviews. And all of this is to try and, for instance, give clinicians guidelines in terms of what should they be doing to continue taking care of families, and it also helps in terms of payers, because again, if there's a recommendation out there to be able to do this, then payers, insurance companies, in other words, are more likely to pay for that clinical care. So for us, it's a priority in terms of being able to do that. We do want this to be data-driven, though. So one of the things we're going to need to do, and Carol and I talked about this yesterday, is actually, as we're harmonizing the data, the good thing about being able to partner with the families, and correct me if I'm wrong, Carol, about this, is that the families have been good about being able to say, I'm this person in this manuscript or in this paper that's published, so we'll be able to have an accounting for all of that. We know for the people who are in Simon's VIP, we know their names because they've registered in this, and that way we can harmonize all of this and make sure that we're not double counting or double dipping. And it's especially challenging, as I'll show you in a second, because in many cases the mutations are exactly the same. The ages and the genders, depending on how old people were when they were in the publications, might be the same, so we can't really intuit this just by looking at the data that we have. We really are going to need the individually identifying information. Okay, so for Simon's VIP, knowing that at least at this point, based on what we know, the ceiling on this could be 64, the total number of participants could be 64, we currently have 38 that are registered in Simon's VIP. So what that means is we've got a chunk of people. It's great, it means we've got a lot of people registered, but also means we have a chunk of people who we don't have registered, and once we do this harmonization with what's in the published literature, then we'll know for sure what that gap is between the 64. So in Simon's VIP, one of the things we are actively doing right now is, I was worried that some of this gap was simply a language issue, that right now we have this in English as an example, and considering this is an international group, I was worried that we didn't have enough languages represented. So we currently actually are in the process of, we've picked the 5 most common languages by surveying the families, that we will have the consent at least in the basic information translated into. The website won't be translated, I apologize about that, but just to be able to participate, the consents will be translated. We'll have translators on the phone to be able to do the medical history interview and the Vineland assessment. And that way we can at least remove that barrier, I hope. So what we need Carol and the families to help us do is to get this, eventually this 38, as close to the 64 as we can. The other portion of this is that of the 38 that are registered, we only have 22 that actually have data in the system. And I know this gets confusing about, well, I consented, am I finished? And the answer is no, you're not. Now the fun begins. And so we need to really be able, and hopefully as we're talking about this to the families, we can help them to understand and get the word out in terms of this. This is really, we've skinnied it down in terms of the medical history interview, the Vineland assessment, for those of you who have done it, somewhere depending on the complexity of your individual family family member between 60 and 90 minutes, but we really have tried to streamline this. And Ashley Wilson, who's the genetic counselor who does this on the phone, I think is lovely. So she, tries to be very accommodating with scheduling, even around parts of the world. She stays very late for Australia, and we do all sorts of things. So anyway, but this is the update in terms of who we've got. As you'll notice, and I'll just show you even more, this is the age distribution of at least who we have in Simon's VIP right now, and not surprisingly, you'll see the skew is towards the younger age. I'm actually excited that our age is getting younger and younger in a very good way, so we're getting even before the first birthday in some cases for this. And so this is for, as we're thinking about therapeutics, I think it gives us opportunities in terms of thinking about very, very early on postnatally, and I would even say it's possible that prenatally will be an opportunity at some point. On the other hand, you'll notice that our oldest individuals are only 25, and so some of the limitations for our families and guidance is we still don't have a lot of long-term outcome data in terms of this, and that really is, I continue to work, and for any of you who think about it, I continue to work on many of our adult populations, trying to get them re-engaged with testing. The limitation is that, to be honest, it doesn't help them as much as it helps the younger generation, but hopefully by getting that information, it's something that we can come back to. And this is really, for many of the molecular biologists, probably one of the most interesting slides. And again, these will all be posted, or we can print these or distribute them, however you want to do this. On the right are the mutations that we know of in the literature. On the left here are the ones that we know about in Simon's VIP. It hasn't changed dramatically in terms of the overall gestalt of this. So 198 is still our most common mutation, accounting for over half. On the other hand, 200 is one of, and I'll talk about it in just a second, 200 is our next most common. And there definitely is the beginnings of an emergence of a phenotype-genotype correlation. I'll just say it's the very beginnings of an emergence. Certainly the families can chime in. 200 looks to be less burdensome than 198, but in terms of, and there is one nonsense mutation, and that definitely is the mildest of the mutations that we've seen. But anyway, in terms of this, we are still clustering. There are the hot spots based on at least my meager understanding of the biology, the three-dimensional structure of the protein, everything else. I think this makes sense in terms of these being pathogenic. The other good thing is that from the diagnostic laboratory point of view, everyone I think now recognizes this, and so these are no longer being reported as variants of uncertain significance. Raid and Kim, tell me if you think differently, but now from my point of view, they're being reported as pathogenic, which in a good way I think is helpful for the families to appreciate the diagnosis and the clinicians. So this is for the clinicians. This is really the money slide, I think, in terms of summarizing most of the clinical data by frequency. And again, all of this is available to the families if the type on this is too small. The consistent thing that we have, we call it penetrance is the word we use, but essentially what percentage of individuals with a genetic difference have the feature. 100% of individuals have some sort of neurodevelopmental issue. Again, I'll show you in a second, it changes by severity, but that clearly is what we see in 100% of individuals. Next most common is actually eye differences. And on the right, I've spelled out, and again, I'll put it in bigger font in a second, but eye problems, whether it's nystagmus, nearsightedness, astigmatism, but eye problems are another sort of common feature. And so again, at least when we counsel the families, it's something we keep a very close eye on, because if we haven't correctly diagnosed the eye problems, it ends up being a handicap in terms of fine motor skills, learning, development, and so we want to make sure we've taken care of that. Next most common, and I would say these are important, but not for different individuals, differentially impactful, but gastrointestinal issues, constipation, diarrhea, alternating constipation and diarrhea, problems in terms of failure to thrive, short stature, especially more difficult early on. And then the other things I would say are relatively less common. We see some of the usual things in terms of ear infections and fluid in the ear and things like that that may cause some hearing problems and may compound some language issues. But thankfully, we aren't seeing a lot of either birth defects, a lot of later functional problems in terms of kidney, heart, cancer, diabetes, any of those things, we're not seeing them very, very commonly. And I'll get to seizures in particular in just a second. But this is primarily the good news for families is I'm not seeing this as a medically extremely burdensome. It's really mostly neurodevelopmentally, behaviorally, the things where we see the burden in the eyes. So in terms of the different developmental diagnoses, again, everyone's carrying at least one of these diagnoses. I will emphasize to the families that autism is a little bit sort of fuzzy around the edges. So there's some individuals that have an official diagnosis of autism. There are other individuals that don't carry an official diagnosis of autism, but they have some behavioral features that overlap with autism. Within this, to me, it's a bit of a gray zone, and I think people are doing what they need to do to make sure that their children are as productive as possible, and they get ABA therapy or whatever behavioral intervention they need to do that. But in general, we are having some other issues in terms of attention deficit disorders, some coordination disorders, some of the things that we see very typically with children that have these issues. One of the big messages to the families, and I think I've made this point before, is that language is certainly a bottleneck early on, being able, expressive language in particular, but do not undersell your kids in terms of their comprehensive understanding, their receptive language. They understand a lot more than they're able to say, and this is true of individuals of all ages, so don't sell them short in terms of that. And we have a lot of methodologies, whether it's literally productive language with speech, communication devices, basically iPads that we've souped up for communication devices, lots of things to allow the kids to communicate better, and I think they're now showing us exactly how much they can do, and it really is remarkable. I'll also say don't give up, especially with the young children. I do think we're starting to see evidence that very early aggressive intervention, so as we start making these diagnoses in 3-week-olds or children that are a few months old, I think we're in a good way getting in very early and helping in terms of a lot of that language. The one other thing I'll say, which is just a tip mainly, again, to the parents that are listening, try and think about and work with your therapist in using multiple ways of being able to access the brain. So just one example of this, some of the kids actually are very either musically inclined or like music a lot. And one of the things we found in terms of doing that is with their language development, actually a lot of singing, being able to learn to speak by learning the lyrics of tunes and being able to put together the part of the brain that's involved in music and being able to bring that also to bear in terms of the language centers. So like I said, lots of different things coming up, and as I always tell parents, there are a lot of technologies that are incredibly useful to support our kiddos, so don't sell them short. This is what we've done in terms of the Vineland assessment. So as an example, the Vineland is a measure of adaptive behavior, and with this is really how kids do in terms of day-to-day functioning. I actually think it's very important. I would argue in some cases more important than an IQ, because it's what it takes to actually get through life on a day-to-day basis. I've color-coded really the 2 most common mutations that we've seen, and again, it's limited numbers, and that's why I'm encouraging in part the families, even if they've been published in the literature, they haven't all gone through the same assessment of a Vineland assessment, and I do think it's very helpful for us to prognosticate and get some ideas about some setting of expectations. What you can see in blue are the 2 individuals that have the 200 mutation, and again, it's not a completely bimodal distribution to be able to see this, but I do think the sort of gestalt that we have is that the 200 mutation tends to be a little bit better in terms of function, and then again, in large part, it's because of what our numbers are, but 198 is having more issues. I haven't put into this the age of all the individuals. I put in some of the age of milestones, but not their chronological age right now. I do think that the earlier, again, we make the diagnosis, whether it's a genetic diagnosis and or a neurodevelopmental diagnosis, I do think that early intervention helps to prop that up. So I think some of our older kids didn't all get that advantage very, very early on. But this is what we've got in terms of that. In terms of how to interpret the numerical score on a Vineland, it's similar to an IQ score in that the mean in the population is 100 and a standard deviation of 15. So as you can see, all of our kiddos are below average in the sense that average is 100, but if this were an IQ, which it's not, but if it were an IQ, we'd say less than 70, for instance, would be someone who are intellectually disabled, and then again, the farther below that, the more challenging. But this is in general a summary in terms of how we're doing. So I think there are 2 diagnoses, right? There's a clinical phenotypic diagnosis, I would argue, and then there's a genetic diagnosis. That this is the one outlier for those of you, again, who think about the biochemistry and molecular biology. I'm not sure the way this works is the same thing, right? So, in other words, if we wanted to describe the clinical phenotype, that might be a different disorder, in fact. I think I still worry that this could be the cause of this child's neurodevelopmental issues, but it may not be if we want to describe this syndrome, what we would put in with the rest of the kiddos with missense mutations. And all of these, I should say, are de novos, so it fits in with what we've talked about before. Okay, so just very, very briefly, in terms of some of the things that we see from a medical point of view, and I've highlighted here, macrocephaly, or a larger head size, is something that we consistently see. It's not 100% as you'll see, because the N on this is 22 in these slides, but again, it's relatively consistent. That just means a head size 2 standard deviations or more above the mean. So macrocephaly consistent, and again, something that we see with other neurodevelopmental disorders. Epilepsy, and again, the sample size on this is 22, so we still have, it's less than a majority of individuals with epilepsy and seizures. I'll get into medications that have been working in a second. They don't all present in the neonatal period, so it is something we need to continue to be attuned for, and as you can see, they can be everything from, sort of a tonic-clonic grand mal seizure, but also some absence seizures, so it is something for parents if you're concerned about. I always say take a video, show it to your neurologist, show it to your pediatrician to make sure you get the EEG. The numbers are very small, so I don't want to say that we have great evidence that it works, but empirically from what the parents are telling us, Onfi seems to be a medication for epilepsy that seems to be working better than some of the others. Whether or not this pans out in the long run, I think we need to see, but that's part of what we're trying to get from families is I call it news you can use, but practical information from each other about what therapies seem to be working better or worse. And so this is, again, something we're going to be working, it's one of our priorities in terms of getting this information. I use the current medications to be a way for me to assess behaviors that are important enough to require medication. So in other words, burdensome enough. So within this, besides seizures, there are also some issues with anxiety in some individuals, so anxiolytics, some sleep issues, and within this, a couple of our young adults who have had more problems with psychosis and needed some medication, psychiatric medication for that. So in terms, and I'll just wrap up here and then pass it over to Rada, but again, predominantly a neurodevelopmental, neurological condition as we're seeing here. And I think the biggest thing for us is that within this, we're also going to have, besides what I'll call this cross-sectional data, longitudinal data. So for everyone who's in VIP, at least what we're trying to do is relatively minimally burdensome, but once a year come back for half an hour is what we're trying to make the burden, be able to go back and see if there are changes over time. So we actually can see the same person and see how they're evolving and developing. Okay, quick questions before we go on? Joe.
So, Wendy, one of the things that we are contemplating, and those of you who visit the Facebook page once in a while, you'll see there's starting to be this last few weeks, so this steady clamor and desire on the part of the families to have another family conference. And what just occurred to me, maybe one of the best ways that we can get that number from 38 to 64, or maybe higher by then, is to actually go ahead and do that and incorporate that into the family conference so the interviews can be done, when you have a captive audience. But I wanted to see if that seemed to make sense, because we're sort of contemplating that, and it's now making more sense to me because of the need to have that additional data.
Well, I think for sure, when we bring the families together, it's a good opportunity to do research in general, right? Like you said, it's a captive audience, so whether it's our baseline, whether it's our follow-up in terms of the families, either one is fine. The one thing I'm just cognizant of is that we do try and make it so that families that can't travel for whatever reason can still do this by telephone, online in terms of doing this. And I think we have also a fire under us to get something in gene reviews as quickly as possible, because that immediately impacts families. So I'm not sure what our timeline ends up being for the family meeting, just because it's logistically, it takes booking hotels and things like that. I hope we don't slow down whatever the sample size is based on the publication from getting that into gene reviews, and then we can continue to expand this for other things going forward.
So we need to drive sooner.
So I think both are good.